So I want to start up where we left off yesterday. It may be a review for some of you, but I think it's important to make sure there's a clear explanation of what's going on with Rosalind Franklin. Rosalind Franklin was a woman who was hired at King's College in the early 1950s, and she was told that she would be the head of the department. And that was what was so enticing to her, was that she would be the head of the department. Another man in the department thought he was going to be promoted to that position. And when Rosalind Franklin arrived, he was on vacation. And when he got back, she had already changed the lab to suit her very specific tastes. And she was already running things and already running experiments. And she had already started working with the graduate student that was in the department and mentoring that graduate student. So this gentleman who was in the department thought that kind of felt pushed to the side, and that made him rather bitter and kind of angry towards Rosalind Franklin, which is why later he would slip photo 51 to Watson and basically undermine all of Rosalind Franklin's work. Rosalind Franklin specialized in X-ray crystallography, which is basically the practice of taking X-ray pictures of molecules, and she uncovered DNA's structure. She was able to see that very characteristic double helix shape, and that's what this X right here is showing you. It's showing a twist in the DNA molecule. Watson and Crick were working at a rival university, and they were also trying to figure out DNA structure, and they were getting nowhere. They had put forth a hypothesized DNA model. Rosalind Franklin came in to check it. She disagreed with their conclusions, and it kind of halted their work on the DNA model because they were just on the wrong track, and the head of their department didn't wish them to continue. The thing about figuring out DNA structure is that it was going to make whoever figured it out famous. They would win a Nobel Prize. It was the thing to figure out in biological science at the time. So Watson and Crick were desperate to figure it out. Franklin was miles ahead of them. She had already taken photograph 51 at this point, and she was pretty sure that DNA was a double helix. She was so dissatisfied with the way she was being treated at her university that she decided to quit. The university insisted that she finish the paper that she had started, and so she wrote up her paper describing DNA's structure based on her evidence from photo 51. Watson and Crick were slipped a copy, showed a image of photo 51 by the other man in Franklin's department. They quickly sketched that image and took it back to their lab and started constructing their DNA model. Now, I want to be clear, Watson and Crick did do some work. They figured out the way DNA was arranged, and they were able, from photo 51, to work out all of DNA's structure. But they wouldn't have been able to do that without Rosalind Franklin's work. The last really bad thing that happened to Rosalind Franklin concerning this was that her paper was altered without her knowledge or permission. So the journal that published both Rosalind Franklin's paper and Watson and Crick's paper published them in the same issue. They placed Watson and Crick's paper before Rosalind Franklin's, and it's important to understand that the way the papers were arranged was important at the time. You would arrange the most important papers first. You would also arrange the ones that had supporting evidence after the initial paper. So they were already situating Rosalind Franklin's paper as the minor paper with supporting evidence, just based on the position in the journal. The next thing they did was they added a line to her paper that said that her conclusions were concurrent or supported the conclusions of Watson and Crick, as stated in their paper. That one sentence changed her work, which was done earlier, so it should have been considered more prominent because it was discovered first, into simply a supporting argument for Watson and Crick's discovery, instead of a discovery in its own right, as it should have been recognized. The reason I think it's important to discuss Rosalind Franklin and to make sure it's clear about what happened to her is that we often think of science as being at the forefront of culture and technology, and in many ways it is. But there are definite issues that still remain concerning science treatment of women and minorities. Even today, as a woman, you are 50% less likely to be published, regardless of your findings in your paper, and the same holds true for minorities. If your name is not classically European, you are 50% less likely to be published. And if we don't recognize our history of sexism and discrimination, we can't move forward and do better. So I think it's very important to give Rosalind Franklin the credit she is due, because she died never knowing her contribution to DNA. She died shortly after her paper was published due to radiation poisoning as a result of her work, so she didn't even know what she had contributed. And that's why I want to make sure that we talk about her and everyone understands the great contribution she made to science. So we're going to talk about DNA structure. We're going to talk about how it's a double helix. Now, the common way to discuss this is calling it a twisted ladder or a spiral staircase. But really all that means is that there are two backbones. There are two sides to DNA. It's composed of nucleotides that are linked together. The nucleotide was first discovered in 1920 by Levine, and he characterized it as the subunit of nucleic acids. A nucleotide contains a 5-carbon sugar, 1 phosphate group, and a nitrogenous base. There are 2 types of nucleic acids found in living organisms, DNA and RNA. Each has a unique nucleotide. The 4 nitrogenous bases for DNA are adenine, guanine, cytosine, and thymine. This is what the nucleotide looks like. We have the phosphate group here. It is often represented as a circle. Then we have the 5-carbon sugar. That means there's a carbon at each corner here. In DNA, the sugar is deoxyribose, and that is important to know. And then we have the nitrogenous base. Please make sure that you get a copy of that you know how to draw the nucleotide. That will come up later as well.